Thank you, Ms. Samarias, for having me. Now it's time to start our today's conference third session, and this session will contain talks from the field of cancer biology, natural product, nanotechnology, and parasitology. Now, I would like to invite our first speaker, Dr. Madiha Arshad from University of Education, and she will talk about evaluation of antibacterial activity of arsenic nanoparticles produced by potato against Staphylococcus aureus. Please, Dr. Madiha.
Assalamu alaikum. Welcome all in 7th International Conference on Applied Zoology 2024. This is me, Dr. Madiha Arshad from University of Education, Faisalabad campus. The topic of today's talk is evaluation of antibacterial activity of arsenic nanoparticle produced by potato Solanum tuberosum against Staphylococcus aureus. First of all, what is toxicology? Toxicology is the branch of science which deals with the toxicants and poisons and their effects and treatments. While a toxicant is a substance that can produce deleterious conditions on a living system, and they may be man-made or naturally occurring. There are several categories of toxicants, but the general toxicants are drugs, radiations, chemicals, heavy metals, and infectious agents. Some of the environmental toxicants can pollute air, they can pollute biologically water, and pesticides and indoor fumes by biomass combustions can also serve as environmental toxicants. Arsenic. Arsenic is an element that naturally occurs in the earth crust. It can be present in plants, soil, sediments, airs, various foods, or drinking water, and anthropogenic procedures can also add arsenic to our environment. Arsenic is a transitional metal and have 4 oxidation states, can make covalent bond with other metals. While it is steel gray in color, brittle and solid, tasteless and odorless, and less thermal and electrical conductor, exhibit shine and glow. There are some benefits of arsenic and as well as some disadvantages. The benefits are as follows: as a treatment of diseases such as diabetes, psoriasis, syphilis, skin ulcers. It can also use as wood preservative, glass making industries. Its applications are in the mining, metallurgical, and semiconductor industries, and agricultural chemicals as well. While it can change the behavior of any animal, long-term storage can delays decompositions and toxic to the plants and animals as well. Arsenic can be exposed via dermal contact, inhalation, absorption through skin, and gastrointestinal tract. Arsenic causes oxidative damage, which can cause chromosomal aberrations and interference with cellular signaling. It also helps in activation of transcription factor, which can alter the gene expression, and all these factors leads to the carcinogenicity. The main objective of this research was to synthesize the arsenic nanoparticles from potato peel extract, its characterization via UV visible spectrophotometry, FTIR, and XRD, and also to evaluate the antimicrobial effects of arsenic nanoparticles against Staphylococcus aureus. Significance of this study is to synthesize arsenic nanoparticles utilizing plants extracts, and that can offer several advantages over physical and chemical approaches like cost-effective and environmental friendly. Arsenic nanoparticles are less prone to inducing bacterial resistance compared to antibiotics, due to their direct interaction with the bacterial cell wall, obviating the need for penetration. Methodology. Materials used in this research was sodium arsenide, distilled water, potato peel, Nutri-Tiger, ethanol, standard antibiotic, gentamicin. Plant was collected from a Yube Agricultural Research. Sodium arsenide salt was purchased from Sigma Eltridge. Stock solution of sodium arsenide of 0.01 molar was prepared. Arsenic nanoparticles were synthesized by adding 500 ml plant extract in 500 ml of salt stock solution with a ratio of 1:1. After that, it was heated at 80 degrees centigrade under vigorous stirring using a magnetic stirrer. The reaction mixture changed into dark brown and the mixture was allowed for 30 minutes for a complete mixing of salt solution. Later on, precipitates were separated from the reaction mixture by centrifugation at 6,000 RPM for 20 minutes and dried in Petri plates at 60 degrees in the oven. Finally, arsenic nanoparticles were collected for experimental work. Characterization was done using UV visible spectroscopy, FTIR, and XRD. While antimicrobial activity analysis of arsenic nanoparticle was done using pathogenic bacterial strain of Staphylococcus aureum, which is gram-positive bacteria, and it was used to test the efficacy of arsenic nanoparticles against them. Those were prepared at different concentrations of arsenic nanoparticles like 10 mg per ml, 20, 30, 40, and 50 mg per ml in respective dose groups. Well diffusion method was used to analyze the minimum inhibitory concentration. Staphylococcus aureus overnight cultures were diluted with nutrient broth. Stock solutions of arsenic nanoparticles for MIC were produced separately. Different concentrations of arsenic nanoparticles were introduced to tubes holding test organisms. Positive and negative controls were made individually with or without arsenic nanoparticles and organisms. The test tubes were placed in an incubator and incubated overnight at 37 degrees centigrade. Turbidity was measured to assess the growth of an organism in the presence of a substance, and the lowest dose that inhibited the organism's growth was designated as MIC. Calculation of minimum inhibitory concentration. Statistical analysis was done using SPSS version 22.0. Results. UV visible absorption spectroscopy gives the absorption at 230 nanometer, that was 3.18 nanometer. This is the graph showing the UV visible range at 3.018. FTIR graph shows that the broad peak seen at 335.34 per centimeter represent OH stretching in phenolic compounds. Sharp and strong peaks observed at 2924.69 centimeter indicate CH stretching in aromatic compound. Sharp and strong peak seen at 1579.68 shown nitrogen oxide stretching in the nitro compound. A weak peak observed in 1390.26 per centimeter indicated C double bond or stretching carboxylic acid, and strong peaks seen at 1118.55 centimeter correspond to C bond C stretching the vibrations and C bond H stretching in aliphatic amines respectively. This is the peaks of different phenolic and oxide groups. To detect the existence of arsenic nanoparticle samples was crystalline and X-ray diffraction to investigate the structure of nanoparticles. In this figure, the XRD pattern that was created exhibit four peak values 28.35, 34.27, 40.52, and 44.21 of pure metallic arsenic nanoparticles. Scherer formula calculated that crystal had a D size of 35.81 nanometer. Using this Scherer formula, we can measure the size of the particles formed from potato peel. Antibacterial activity of arsenic nanoparticle against Staphylococcus aureus. The impact of arsenic nanoparticle on the growth of Staphylococcus aureus was investigated through 7 different groups. Results revealed a significant reduction in bacterial growth compared to control group with increasing mean values observed across the treated groups. This suggests that arsenic nanoparticles exert inhibitory effects on the growth of Staphylococcus aureus in a dose-dependent manner. These findings underscore the potential of arsenic nanoparticle as antimicrobial agent, highlighting their relevance in combating bacterial infections and offering avenues for the further exploration in medical and environmental contexts. Here are the mean and standard error of mean of different control groups, which is clearly significantly different from the control groups. Here in this figure, antibacterial activity of biogenically synthesized and arsenic nanoparticles against Staphylococcus aureus is revealed. Determination of minimum inhibitory concentration. Minimum inhibitory concentration falls between 500 microgram per ml and 250 microgram per ml. MIC is equal to 500 plus 250 divided by 2, that is 375 microgram per ml. MIC value of arsenic nanoparticle was between 500 microgram per ml and 250 microgram per ml, which is calculated as 350 microgram per ml. Conclusion: the utilization of aqueous extract of potato peels in the production of arsenic nanoparticles proved to be a successful approach. The current study introduced a very effective environmentally sustainable procedure for synthesizing smaller sized arsenic nanoparticle. And from this research, it can be concluded that arsenic nanoparticles can be synthesized from potato peel, and these are useful against pathogenic strains of Staphylococcus aureus. In this research, it seems that biogenic synthesis from a potato peel is possible, which is a cheaper way to produce arsenic nanoparticles. It can be recommended from the present study that arsenic nanoparticle can help explore the new way to use arsenic nanoparticles as antibacterial agents in future endeavors. Thank you for your attention.
Dr. Madiha, for such an informative talk. Now, I would like to invite our next speaker, Dr. Sabah Farouk from Allama Iqbal Open University, and she will talk about evaluation of frequency of Acinetobacter baumannii from surgical site infection and assessment of multiple antibiotic resistance index. Please, Dr. Sabah.
Bismillahirrahmanirrahim. I am really thankful to the organizing committee of 7th International Conference on Applied Zoology and in collaboration with the University of Sargodha for providing me this opportunity to share my thoughts on this esteemed forum. I am Dr. Sabah Farooq from the Department of Biology, Allama Iqbal Open University, and today I am going to discuss evaluation of frequency of Acinetobacter from surgical site infections and assessment of multiple antibiotic resistance index. The presentation includes introduction, objective of the study, methodology, results, and conclusion. Acinetobacter baumannii is a gram-negative organism which is widely distributed in environment. It is found in soil and water as well. It is an opportunistic pathogen in human which is responsible for nosocomial infections, This organism is rapidly acquiring multi-drug resistance. Multi-drug resistance is the attribute by which bacteria can resist one antibiotic belonging to three different antibiotic groups. And this multi-drug resistance is an emerging threat for therapeutic and healthcare facilities. Nosocomial infections are responsible for hospital-acquired pneumonia, septicemia, meningitis, urinary tract, and wound infections. Objectives of study include isolation and characterization of Acinetobacter baumannii from surgical site infections, identification of bacteria using biochemical and automated profile indexing, profiling of resistance of antibiotics against different antibiotic groups. For this purpose, 165 surgical site infection samples were collected from different healthcare facilities of Islamabad. These samples were subjected to standard microbiological procedures for bacterial isolation. 33 bacterial isolates were obtained, which were subjected to morphological and colony examination, followed by microscopic examination. Based upon gram staining and biochemical characterization, including catalase test, indole test, urease, motility, triple sugar iron utilization test, citrate utilization, oxidase test, 9 bacterial isolates were considered as base. This slide represents the isolation of Acinetobacter baumannii from different surgical site infection samples. Most frequent isolation was from catheter tip, followed by suction pump. Only one isolate was obtained from blood and one isolate was obtained from pus swabs. The growth pattern representative of Acinetobacter baumannii cultivation are shown on blood agar plate and McConkey agar plate. Biochemical tests carried out for the identification of organism include catalase test, indole test, citrate test, urease test, oxidase test, motility test. All Acinetobacter baumannii isolates were positive for catalase test, negative for indole test, positive for citrate utilization test, negative for urease, negative for oxidase test, and negative for motility test. Further, species level identification of Acinetobacter baumannii was carried out by using API strips, automated profile indexing, and recording all the results of these tests. Identified isolates of Acinetobacter baumannii were then used for antibiotic susceptibility and resistance profiling by Kirby-Pure method. In this process, test diffusion method is used to assess the antibiotic resistance of the test organism by using the zone of inhibition along with the comparison of CLSI standards. The bacteria were classified into resistant, intermediate resistant, or sensitive to the selected antibiotic. After recording the resistance and sensitivity pattern of bacteria against different multiple antibiotics used, MAR index was calculated for all the 9 bacterial isolates. Isolate number 1 was resistant to 9 out of 11 antibiotics used, exhibiting 0.81 MAR index. Second isolate was resistant to 9 out of 10 antibiotics used. The third exhibited resistance to 10 out of 11 antibiotics used. Both gave MAR index of 0.90. Fourth isolate was resistant to 8 out of 11 antibiotics used. Fifth isolate was resistant to 9 out of 11 antibiotics used. Only one isolate gave resistance to all antibiotics used against it, exhibiting MAR index of 1, which is extremely high. Seventh isolate was resistant to 2 antibiotics out of 7 used, and it exhibited the least MAR index. Eighth isolate exhibited 10 out of 11 and 9 also 10 out of 11 resistance to different antibiotics used. Keeping in view the results of study, we can say most Acinetobacter baumannii isolates had MAR index greater than 0.2. This index represents the emergence of these isolates from a high-risk contaminated niche where antibiotics have been extensively used. The isolate, which was resistant to all the antimicrobial agents used against it, was isolated from a catheter tip. It is now the need of hour to regulate the use of antibiotics and to stop the spread of antibiotic resistance across different bacterial genera so that the burden of hospitalization and extensive antibiotic use can be minimized. With the take-home message of regulation of antibiotics at our individual level and national level, I will conclude my presentation. That is all from my side. Thank you very much for listening. Thank you very much for your attention.
Thank you, Dr. Sabah Farooq, for sharing such valuable information. Now, I would like to invite Ms. Rahila Sultana from University of Sargodha, and she will talk about the role of RSTP53 in liver cancer. Please, Ms. Rahila Sultana.
Assalamu alaikum. My name is Rahila Sultana and I am the student of MPhil Zoology in the University of Lahore Sargodha campus. The topic I have selected for my thesis is role of RS1042522 TP53 gene in liver cancer. Liver cancer is a disease in which malignant cell forms in the tissue of the liver. Here you can see the difference between healthy liver and fatty liver, which may lead to fibrosis and then cirrhosis. Many risk factors are presented here, like old age, diabetes, excessive alcohol consumption, chronic infections. There are so many signs like lumps, swollen abdomen, fever, abdominal pain, and fatigue, etc. The main focus of my research and investigation is to investigate the role of TP53 gene polymorphism in liver cancer, to investigate the association of demographic parameters with TP53 gene polymorphism. TP53 gene play an important role to control the DNA damaging and apoptosis. But when mutation occurs, it causes liver cancer. We have selected the blood samples from PKLI from Lahore, and then we proceed our methodology. The methodology of our research is sampling. Then we will collect from PKLI Lahore. Then we extract the DNA. Then we design the primer, DNA amplify, and then bake amplicon conformation. DNA extraction is proceeded by the proper method in the lab of the University of Lahore. Then amplify the DNA. After that, we compare the different demographic parameters in cases control and control in form of parameters in cases and controls of liver. In this table, we will compare the demographic parameters. This study conducts the comparison between age, gender, and smoking status. When age divided into two categories, smaller than 48 and greater than 48, it reveals that age increases the rate of risk and plays a significant role in increasing liver cancer. Gender also plays a significant role in increasing the rate of liver cancer. Smoking status reveals that it also performs a significant role in increasing the rate of liver cancer. In this slide, the second table shows the genetic constitution compared to 25% control. It shows the reference category, which is presented here in the first column. AA wild type reduced the danger of liver tumor, while AG 53%, which shows while GG 30% variant. These hetero and mutant plays a significant role in increasing the risk of liver cancer. Then we conduct the association of TP53 RS1042522 polymorphism with gender. It shows the AG hetero in male plays highly significant association, while in female only AG has the risk of highly significant in liver cancer. There, in this table, we have conduct the association of RS1042522 polymorphism with age. It reveals that patients smaller than 48 shows highly significant in AA wild, AG hetero. On the other hand, the patients greater than 48 only GG mutant shows highly significant in liver cancer. In this table, the association which is shows the polymorphism RS1042522 smoking status. This study reveals that smokers AA wild, AG hetero and GG mutant plays a significant role in liver cancer, while in non-smokers GG mutant plays a significant role in increasing liver cancer. The conclusion of my study is that TP53 is actually an inhibitor, but when it mutated, it caused liver cancer. TP53 plays a significant role in increasing the risk of liver cancer. This can be used as a biomarker to diagnose liver cancer in early stage. In this slide, we have to show that recommendations. If I want to study the more on the TP53 gene, then it will move towards integration of genetic testing, focusing on age, main and gender main research. It also shows the smoking cessation programs, public health education, and multidisciplinary approach. Thank you so much.
Thank you, Ms. Rahila Sultana, for such an productive talk. Now, I would like to invite our next speaker, Ms. Areej Seher from University of Agriculture, Faisalabad, and she will talk about neurotoxic and nephrotoxic effects of heavy metals burden in black cards, a profound insight into urban pollution in Faisalabad, Pakistan. Please, Ms. Areej Seher.
Assalamu alaikum everyone. My name is Arid Seher and I am an MPhil scholar from University of Agriculture Faisalabad. Today I am excited to present my research on neurotoxic and nephrotoxic effects of heavy metals burden in black kites, a profound insight into urban pollution in Faisalabad, Pakistan. My research focuses on the bioaccumulation of heavy metals and the toxicological impacts they have on urban raptors, especially black kites. So I assessed levels of heavy metals in black kites and examined how these metals affect their brain and kidney tissues. As far as we know, urbanization and industrialization are major reasons of environmental contamination, especially through heavy metals. These metals are released into the environment through various activities like factory emission, improper waste disposal, and automobile exhaust. Once these metals enter ecosystem, they accumulate in living organisms, posing significant health risks to both wildlife and humans. I studied on black kites because being top predator and scavengers, they are highly exposed to these pollutants. Their top position in the food chain makes them an ideal species to study the impacts of heavy metal concentration on urban wildlife. These are some primary objectives of my study. First, I aim to assess concentration of these selected heavy metals such as lead, zinc, and manganese in brain and kidney tissues of black kites. Secondly, to explore neurotoxic effects of these metals, focusing on their impact on the brain. Additionally, to evaluate and study nephrotoxic effects on the kidney tissues of these birds. So finally, I wanted to highlight the use of black kites as bioindicator. My research focused on two highly polluted sites of Faisalabad. We captured black kites from Ghulam Muhammadabad, considered as residential area, and also collected from traffic area of Janawala Road, Faisalabad. Black kites are abundant in these areas as they often scavenge waste dumps for food. This made Faisalabad the ideal location to study heavy metal pollution. To carry out the study, I dissected brain and kidney of black kites from both polluted areas of Faisalabad, and these tissue samples were chemically digested using nitric acid and hydrogen peroxide in 3:1 concentration. I choose these tissues of kites because they are exposed to toxic effects of heavy metals. The collected samples were analyzed using atomic absorption spectrophotometer at high-tech lab in University of Agriculture, Faisalabad, in order to measure concentration of lead, zinc, and manganese. This method provides us to accurately quantify the heavy metal levels in bird tissues. I also performed histological examination to observe any tissue damage or structural damage caused by the metals. Additionally, behavioral assessments were conducted to detect neurotoxic effects. Then statistical analysis was conducted using ANOVA to show variation significantly or non-significantly. Let's now take a look at findings of this study. This table shows the descriptive analysis data of three metals in kidney and brain tissues of black kites among both selected locations of Faisalabad city. This bar graph clearly elaborates mean levels of zinc, manganese, and lead within selected organs of kites. Here, Site 1 refers to Ghulam Muhammadabad and Site 2 as Janawala Road, Faisalabad. According to graph representation, zinc concentration were the highest, followed by manganese and lead. When comparing the two study sites, Ghulam Muhammadabad exhibited higher zinc concentration in the kidney while higher zinc level in brain at Janawala Road. ANOVA analysis revealed significant variation in the levels of zinc, manganese, and lead between the two polluted sites. This result indicate localized differences in pollution intensity among tissue samples of black kites. This analysis revealed alarming levels or significant level of heavy metal concentration in the black kites. Lead concentration in the brain is strongly linked to neurotoxic effects, including impaired motor coordination. Moving on to neurotoxicity, these birds also exhibited behavioral changes like decreased flight coordination and agility, which are key indicators of neurological impairments. These symptoms are consistent with neurotoxic effects caused by heavy metal poisoning, which disturb normal brain function. Next, I discuss about nephrotoxicity in the kidney tissues. Zinc level was observed highest in the kidney, cause nephrotoxic effects, including tubular damage and impaired renal function. The affected kite showed evidence of tissue necrosis and fibrosis, which are common outcomes of heavy metal toxicity. Manganese exposure in the brain suggested oxidative stress, while in the kidney, it caused renal inflammation. This study confirms the significant bioaccumulation of heavy metals in black kites from polluted areas of Faisalabad with profound neurotoxic and nephrotoxic effects. So, my research demonstrated that urban pollution in Faisalabad is having a serious and detrimental impact on local wildlife, especially black kites. The high levels of heavy metals detected in their brain and kidney tissues have long term consequences for their survival. These findings the critical need for environmental regulatory measures to reduce heavy metal pollution and protect biodiversity in polluted urban environments. Future research could expand on this by studying other wildlife species and developing a comprehensive environmental monitoring program using black kites as bioindicators. Thank you so much for giving attention.
Thank you, Ms. Adib said, for your valuable information. Now, I would like to invite Dr. Patricio, University Católica de Chile, and he will talk about null models for understand intertidal lake approach communities in North Patagonian Beach. Please, Dr. Patricio.
Hello, this is a presentation entitled Null Model for Understanding Intertidal Decoupled Community in North Patagonian Beach, Peyuin 41 South Latitude, Chile. I am Patricio de los Ríos Escalante from the Catholic University of Temuco, Chile. In this slide, you can see the co-authors of this present study. As introduction, the intertidal decouples in continental Chile are characterized by this species training in Peruvian coast along practically all coasts, by coexisting species that have not studied with details with interspecific interactions. In continental Chile, the north coast have similarity with Peruvian coast, areas between 38 and 41 south latitude. There is a kind of transition zone, and at south of 41 south latitude, the species are related with subantarctic ecosystem environment or ecosystems. The objective of the present study is apply neural models for understanding the intertidal decoupled community is random or not random. This means that if the intertidal decoupled community has structured patterns or has not structured patterns. In material method, the study site corresponds to a sandy beach with a small rounded stone called Peyuin Beach. This specific geographical location at 4 kilometers south of Puerto Montt in semi-rural zone that was visited the 24th February 2020 during low tide. Data collection. The field species identification was done in situ based on literature description, and taxonomic identification was confirmed by worms. Random quadrants 10 by 10 centimeters were released in a study site with N and 19 for each site during low tide. It was considered the site of quadrant of the basis that the fast movement of littoral decoupling, and this site quadrant will be more adequate for fast moving species and the irregular conformation of the site on the rounding routes. Manually entered data will be recorded in a field copybook. In this place, you can see a map of study sites and the location within south of Chile and South America. And result discussion. The result revealed that the species association were random, though probably, though many species repeated in many samples, and the sharing result revealed that the species reported don't share niche. This means that the species do not share the ecological niche. In consequence, they are not a specific competition because each species has very specific ecological niche, and it is not necessary the interspecific competition. You can see in this site the mean abundance and the standard deviation of different decapod species observed for study sites. In this plate, you can see the result of different null models. You can see in this table the result of species co-occurrence null model, and you can see that in fixed the species association are random, whereas in fixed equivalent and fixed proportional, the species association is not random. This means the existence of structural patterns. Whereas in niche sharing models, you can see in according to Bianca index that the species don't share ecological niche, and this means the absence of interspecific competition. Yeah. In this plate, you can see the graphs that confirm the result of the last table. You can see at the top and left the result of fixed null models, and in the top and right and the low and left, you can see the other null models of species concurrence. You can see in the last two graphs at the top and right and low and left, the existence of a red line that is very far in comparison to the mean index. Whereas at the right and low, you can see the graphs of the result of nature sharing, and you can see that the red line is very close to the mean observed index and very low value in comparison to the mean observed index. As a discussion, the obtained results are particularly agree in comparison to similar observations for Chilean coast, and it will be explained the oceanographic conditions. Similar results were reported for the intertidal coast of the north of Chile, specifically at 33 latitude in Antofagasta region, for the coupled fauna, as well as intertidal invertebrate community in the north of Chile, at the 23 latitude in Antofagasta, and northern Patagonian coast in 38 latitude. This result they note that in your models, species co-occurrence that the species association are mainly random, and few models reveal the existence of structured patterns, and the result of neural models of nature sharing reveal that the species don't share the ecological niche, and in consequence, there are not interspecific competition between involved species in terms of decoupled fauna and intertidal invertebrate fauna in rocky shores. This is the acknowledgement of the present study. Thank you for your attention.
Thank you, Dr. Pratisho, for your profound talk. Now, I would like to invite our next speaker, Mr. Hassan Raza from University of Punjab, and he will talk about new remains of Suadi from the Dhok Pathan Formation, Hasnot, Punjab, Pakistan. Please, Mr. Hassan Raza.
Bismillahirrahmanirrahim. Assalamu alaikum. Myself Hassan Raza, and today I am here to represent my MPhil research abstract and in the 7th International Conference on Applied Zoology 2024. Here is my abstract. I worked on the paleontology and collected fossils from the Sivalics There are three parts of symbolics. The first one is upper symbolics, and second is middle, and the third one is lower symbolics. The collected fossils are for belong to the middle symbolics. I worked under the supervision of respected Professor Dr. Muhammad Akbar Khan. Here is my abstract. New suet fossils comprising dentation recovered from the Dhok Pathan Formation late Miocene outcrop of Hasnot and the Siwaliks group in Pothohar Plateau of Pakistan. The recovered material shows morphometric similarities with the Eurasian genera Propotamocoharis and Hippopotamodon. Based on these similarities, the material is allocated to the Propotamocoharis hysotrachus and Hippopotamodon sivilensis. Further data on the anatomical character of this species, especially for the molars of Propotamocoharis hysotrachus. The methodology which was used during the research is actually uh, the uh, the basic steps are in which we collected fossils and clean the fossils. After that, identify the fossil and give description to the fossils. In this, the studied material consists of specimens and its detail are provided in. The surface collection method was used to collect the fossils. The collected specimens were prepared and displayed in the Dr. Abu Bakr Fossil Display and Research Center, Institute of Zoology, University of the Punjab, Pakistan. The measurements and nomenclature follow Thang Hai 2005 article, and the measurements have been taken with the digital vernier caliper and the dimension were given in millimeter. Photography has been done with the Canon 60 camera and the figure has been prepared using Adobe Photoshop. Here is the specimen of Propotamocoharis hysotrachus. First of all, I want to discuss the uh, classification of Propotamocoharis hysotrachus is actually they belong to the order Archaeodactyla and suborder Suiform, family Suidae, tribe Dicorocorini, and genus Propotamocoharis, and the species name is Propotamocoharis hysotrachus. Here on the left side is the left mandibular fragment. On the right side is M2 of Propotamocoharis, upper M2. First of all, we would like to discuss the right specimen, upper M2. The description of upper M2 is an isolated upper molar. The paracone and protocone are well preserved, and the metacone and hypocone are partially preserved, where the metacone is more preserved than hypocone. The hypoperconule is low and the protoperconule is highly worn. The crown is low and weakly developed anterior cingulum, and the median valley is wide and open labially, while closed by the proendoconule labially. The second specimen is left mandibular fragment with the M2, M1, and also the root of P4 is present. A weakly developed anterior cingulate is present anterior transversely. The M1 is well preserved with a thick enamel. There are four conates. The protoconate is low and well preserved and is the most worn cusp. The prominent protoperculate is connection with the anterior cingulate, and the metaconate is highly cusp and has large metoconulate. The hyperconulate is large and lies close to this cusp. Both the hyperconate and endoconate are much lower than the protoconate and metaconate. A large hypopostconulate is in connection with the moderately developed and thick posterior cingulate. The median valley is broader lingually and with a weakly developed actoconulate. Second, the M2 description. The M2 is less worn and relatively more preserved. It is broken posterior transversely, resulting in the loss of some part of hypopostconulate and the complete loss of posterior cingulate. Anterior cingulate is thicker labiotransversely and weaker and thinner lingotransversely. The protoconate is slightly anterior than the highest metaconate. The grooves are few but not deep. Rest after morphology is similar to the M1. Here is the another species named as Hippopotamodon sibilans. Hippopotamodon sibilans is also large-sized mammal, suet. There are some large-sized suet include Microstronix, Hippopotamodon sibilans, and Tetraconodon. But the Propotamocoharis hystracus is medium-sized suet. The specimen shown in figure is actually the posterior lobe of molar of Hippopotamodon sibilensis. Here is the conclusion of our abstract. There are two species were identified and described in the thesis. First one is Propotamocoris hystracus and the second is Hippopotamodon sibilensis. These are reported from Dog Pathan Formation of the middle Siwalik. The posterior lobe of molar of Hippopotamodon sibilensis and the mandibular fragment with M1 and M2 with root of P4 of Propotamocoharis acidophilus adds substantial information in anatomical and morphological character and represent the taxonomy.
Nanoparticles on the post-thaw quality of cryopreserved sperm of Columbia libia domestica. Please, Ms. Asma Iqbal.
Hello everyone. I am pleased to present my research on the effect of zinc oxide nanoparticles on the post-thaw quality of cryopreserved pigeon sperms. Cryopreservation is a critical technique in cryopreserving genetic diversity, but it poses challenges like oxidative stress that damage sperm during freezing and thawing. This study explores whether zinc oxide nanoparticles, known for their antioxidant properties, can mitigate this damage, specifically in the domestic pigeon, which scientifically named as Columba livia domestica. The primary objectives of this study were to assess the effects of various zinc oxide nanoparticle concentrations at 50 microgram per ml, 100 microgram per ml, and 200 microgram per ml on sperm motility, plasma membrane integrity, and acrosomal integrity. And the other one is to compare the performance of these zinc oxide nanoparticle treated groups against a control group with no zinc oxide nanoparticle. These matrices are crucial for evaluating the potential of zinc oxide nanoparticles in improving cryopreservation protocols. The experimental design involved collecting semen from 10 male pigeons. Samples were diluted in a lake extender with glycerol and varying concentrations of zinc oxide nanoparticles. After cryopreservation in liquid nitrogen for 1 month, post-thaw sperm was evaluated using phase contrast microscopy for motility, hypoosmotic swelling test for plasma membrane integrity, and Giemsa staining for acrosomal integrity. Zinc oxide nanoparticles were evaluated as cryodamage based on their antioxidant properties, with 3 different concentrations treated against the control group. Role of zinc oxide nanoparticles in cryopreservation. Zinc oxide nanoparticles are hypothesized to reduce oxidative stress during freezing and thawing due to their antioxidant capabilities. Zinc plays a critical role in maintaining sperm integrity, motility, and overall fertility. This makes it a promising additive for sperm preservation during cryopreservation, and our study aimed to verify this through empirical tests. Zinc oxide nanoparticles impact on sperm motility. Before freezing, the group treated with zinc oxide nanoparticle at 100 microgram per ml showed the highest motility rate of 86.33%, significantly outperforming the control group, which exhibited a motility rate of 65%. After thawing, the 100 microgram per ml zinc oxide nanoparticle group maintained the highest motility at 89% compared to 63% in the control group. This substantial improvement highlights the potential of zinc oxide nanoparticles in preserving sperm motility. Zinc oxide nanoparticle impact on acrosomal integrity. Acrosomal integrity is crucial for fertilization potential of sperm. Before freezing, the 100 microgram per ml zinc oxide nanoparticle group showed the best acrosomal integrity at 84.67%, and this was maintained post-thaw at 81.67%. While the control group exhibited significantly lower acrosomal integrity both before freezing, which is 15
areas of Faisalabad. We selected Faisalabad because it's in a textile industrial zone. We can say it's a hub of Pakistan industrial zone, and we compared the pollution level or contamination level of pollutants or heavy metals from rural and urban areas. Second is to check the heavy metal concentration in specific tissues. I selected liver and lungs of black kites to check the contamination level. Third is to identify or to check the effect of lead and copper on liver and lungs. What could be the possible diseases or what could be the possible impact on not only black kites but also in food chain, the other organisms, smaller or higher level, including humans. We selected two sites that I already described, two sites, rural and urban. Rural area is included agricultural land and urban zone is industrial zone. Also it's a higher level of traffic area. We selected 12 kites from rural area and we captured 12 kites from urban area by using bait trap or netting. We put meat and we just pour it with the chloroform, liquid chloroform, so that we can capture them. And after capturing, we digested the samples, and through atomic absorption spectrophotometry we quantified the concentration of lead and copper. The dissection was placed it was taken place in the biodiversity lab of Dr. Nazia Ehsan at Zoology Department, University of Agriculture, Faisalabad. We dissected black kites individually, separated the concerned organs, which were liver and lungs in my case, and I then dried the samples which we just cut it off, and from oven I just dried them, chopped the organs in fine particles or pieces, then digested the sample. The procedure was we took 1 gram of liver and lung samples and placed them in the respective beaker, then added 10 milliliter of nitric acid, put it on a hot plate for 30 minutes and heated. After 30 minutes I added 5 milliliter hydrochloric acid and reheated for 15 minutes on hot plate, and then when thick transparent material was formed, I diluted it with distilled water and raised the volume up to 25 milliliter. For analysis, high-tech lab of University of Agriculture, Faisalabad was selected, and we quantified and assessed the concentration of heavy metals there through atomic absorption spectrophotometry, and it was taken in milligrams per kilograms. For statistical analysis, we found significant results, like there was significant difference between rural and urban areas data. The data which was indicating the rural area was less contaminated than urban area. So results are here that site 2, which was urban area, had more amount of copper and lead, in both cases. So here is the mean concentration of heavy metals in lungs. And when we compare the concentrations from both sides, you can see here site 1 has less amount of copper and lead as compared to site 2. So it means urban area has more polluted environment. Similarly, when we talk about the liver, in liver also both sides have a significant difference. Copper and lead has less amount at site 1, while at site 2 copper and lead is seen more in concentration. That is, there is a significant difference. It is 20.97 and it is 31.77. And there is also seen the difference between liver and lungs, because liver is more toxicated than lungs. The effect which we have, some kites has liver inflammation, some had abnormal growth of tissues or cells, which leads to cancer and also liver cirrhosis. And if we talk about lungs, then there were also necrosis and pulmonary fibrosis. We saw that there is a respiratory damage. What could be the possible future directions? We can work on molecular biomarkers that are indicators for heavy metal toxicity in the environment, in any metropolitan city, in any polluted area, or any area which you can understand that this can be polluted with some heavy metals, nanoparticles, or any kind of pollutants. Oxidative stress pathways. We can work on immune response, genomic adaptations, and analysis of biochemical changes in living bodies. And these living bodies can also include human beings. And what is the impact on human health? Because raptors are at a higher level and comes food chain, so they are best bioindicator of the environment and what is going on in the atmosphere, what kind of pollutants are present in atmosphere. So we can describe it more accurately with the study of raptors, that the environment is suitable for living organisms or not, including humans, because in Faisalabad, as we have reported many international level or at national level, that in Faisalabad most of the people has hepatitis, and hepatitis is due to polluted water, as it's a waterborne disease. So polluted water is also due to the industrialization. So this is all about our environment which is polluted, and we describe it in a certain way with the help of raptor studies, that is black kite. Thank you so much.
Thank you, Ms. Hafsa Bird, for such invaluable insights. Now it's time to move towards our fourth session, which will contain talk from the fields of nutrition, toxicology, diabetes, and genetics. Now, I would request our next moderator to join us and continue this fourth session.